Thank you, Angela, and thank you for inviting me to present our work from the Hudson Monash program and also scheduling at a Australia-friendly time. So I'll go ahead and get started. We've been working on establishing a pediatric brain cancer dependency map, and some of you have heard our group talk on this in the past few years. So I'll keep this brief. By way of background, as you're all aware, we've been approaching children in kind of the adult lens, and this has been the case hundreds of years ago, with taking an example from classical painting. And fast forward about 500 years later, and it's only really now that we're realizing that, in fact, childhood malignancies are a different entity compared to the adult. This statement coming out in the past few years from the National Academy of Science really highlights that we need to recognize that children's cancer, particularly children's brain cancer, is a distinct biological entity and harbors unique genetic alterations. And the way that our group approaches childhood brain cancer is really through an integrative genomic approach. And this involves utilizing the childhood cancer genome anatomy projects that CBTN, TCGA, and St. Jude's and other have been leading in terms of providing the landscape of the childhood brain cancer, characterizing the amplification, mutations, translocations, epigenetic alterations, and really marrying that information with functional approaches, namely drug screens and genetic perturbation screens, to identify which of these alterations might be targetable and therapeutic vulnerabilities. And I think, particularly in pediatric cancer, having trained at the Broad Institute and screen some of the adult cancer cell lines, it's really, I think, clear to the community that we need to develop better models of pediatric cancers that really recapitulate the genetic alterations that are found in the tumors themselves. And that's where we've been really working in the last few years, having established this pediatric precision medicine program, really working at the functional element of pediatric medicine. And what that really means is generating models and working with other groups within CBTN and around the world to build a collection of models that we can then interrogate using functional approaches. At the ground level, we've established a robust pipeline for collecting and generating models across Australia, and these are then entered into our living biobank, stored and managed along with the metadata from the patient themselves. At the ground level, I think, we've already had a few successes that illustrate the importance of generating these models. Most recently, we've developed a novel model for a rare form of pediatric gliomas that harbors a fusion that has just very recently been identified by our group and others, which essentially encodes a fusion of the BRD4 LUTX lineage transcription factor. We're currently working with the MNP 2.0 program, which has identified a methylation specific signature that harbors this fusion. And by generating the model in-house from our patient, we've shown that this glioma subtype is actually very sensitive to BET inhibitors, which I think really offers hope for this molecularly defined glioma class. But taking a step back, we really want to generate models across statistically powered groups where we can start to look at genotype-phenotype correlations, and that really requires establishing large cohorts of models. So as Angela mentioned, I think, we've been very grateful to this CBTN network in terms of our ability to work with both the team at CHOP and other groups as well that have contributed, particularly Hopkins, Daytona, and others, and they're shown here, and really represent the largest cohort of different pediatric brain cancers that have entered into our cell line atlas. And you can see we have over 200 different models. And you can see the distribution of our brain cancer models shown here. We have about 180 pediatric brain cancer models, most of these in DIPG, DMG cohort, ATRTs, hemispheric gliomas, ETMRs, and a few of the rarer subtypes of brain cancers, along with 17 fibroblast lines that were established directly from brain cancer biopsies. And I think the importance of having these kinds of numbers is really being able to then do specific genotype, phenotype type analyses along the molecularly defined subtypes of pediatric brain cancers. And just to illustrate, I'm showing here an oncoplot of the high-grade gliomas that are in our collections. And you can see it really spans the different histone variants, H3.1, H3.3. We have wild-type gliomas. We have the G34R variants along with different molecular defined subclasses. And this allows us then to begin to look across these different subtypes and ask which drugs or which genetic knockouts might particularly be impactful in specific histone variant gliomas, additional accessory mutations such as PIK3CA and F1 across amplified genes, or even across different lineages such as adult gliomas versus pediatric. So what do we do in terms of our workflow is we conduct both molecular profiling using whole genome sequencing, transcriptomics, epigenomic analyses using DNA methylomerase and attack sequencing. And we've also recently added total and both phosphoproteome analysis. We then also perform screens, both CRISPR genetic knockout screens to identify therapeutic vulnerabilities, and also drug screens using 3 separate libraries that comprise about 2,000 drugs. This bar graph just shows our progress to date across our 200 cell lines. And you can see that we made fairly good progress in terms of the molecular profiling. CRISPR screens, proteomics, and ATAC-seq have just come online in the last year or so, but we should be ramping up. And really the ability to use this data is, I think, lies in our ability to do multi-omic type of integrative analyses, and we've started to do that. So this is just to show you a multi-omic factor analysis of the pediatric brain cancer lines, where we're now doing tumor subtyping, not just across one particular variable, but across all the molecular features that are shown here. And what we're really finding is that methylation and RNA transcriptomics really are the main drivers of being able to subtype pediatric cancer lines. And you can see that in this principal component analysis type plot, where you can see a nice segregation of DMGs compared to the other gliomas, medulloblastomas, ATRTs, the fibroblast line, and sarcomas. And this really takes into account all the molecular features. So at a functional level, we're interested, we're using these models at the acute level to work with our clinical colleagues to look at particular drug combinations or dose testing for patients where we generate models in real time. But really our main effort is around identifying the next generation of precision oncology agents using these models, and that requires drug discovery and drug development efforts. So to take you through our drug screening pipeline, for all the cell lines that we generate or bring in-house, we screen them through 3 libraries focusing on kinases, a Cambridge Oncology Library, and then a larger library of all FDA-approved agents. We conduct a cell proliferation screen. We've also joined in the past couple of years a worldwide effort called COMPASS, which is consortium of different programs around the world doing ex vivo drug screens in pediatric cancer. You can see the members here, and this is really to harmonize our efforts so that we make sure that we're all doing the screens in the same way. So what we found this far across the 130 or so cell lines that we screened is that actually the majority of drugs do not hit any of the cell lines. So you can see here more than 600 drugs from these libraries are inactive. Another percentage of, actually quite a large percentage of drugs also score quite sparingly in these cell lines. And that has led us now to develop a second generation drug library that is really focused on pediatric cancer. So this is about 750 compounds taken from this data, along with other more clinically active drugs that we know of. And that's really focused on pediatric brain cancer. I'm happy to share that list with anyone who may be interested in that, and also get feedback if we're missing anything. Overall, you can see across these different cohorts that all of the different lineages show about the same fraction of hits, about on average 200 hits or so. And as you can see here, that across the different lineages as well, we don't see any real clustering based on the drug hits. So really the lineages themselves or the types of cancers don't really predict activity. And that led us to use more sophisticated approaches, particularly machine-based learning. And then we've also started to play around with neural network type approaches in collaboration with the Broad Institute to start to look whether we can use our different omic features to predict drug response. And without going too much into the bioinformatics, just focus here on this quadrant, which are those drugs which show the greatest level of correlation to a molecular predictive feature and the least amount of error. And that's shown here in this quadrant. And if we take this quadrant and start to look at the individual drugs, what we can see is at a high level is that the main predictive features are actually centered around the mRNA or transcriptomics, shown here in the dark red, and in lighter pink color. You can see there are some drugs that are predicted more so by the methylation array, and these tend to be the two most predictive features of drug response. Now, if we look at a more granular level, you can see indeed in a couple of cases, this tends to be the case. So for example, EGFR, expression of EGFR serves as a good predictive marker for the response to a number of targeted agents in this pathway. Interestingly, with the PIK3CA, there's actually an inverse response to inhibitors in the pathway, and you can see that higher PIK3A, or I should say lower expression of PIK3A predicts response to inhibitors in the pathway, which is interesting, and it may suggest that there's a kind of a cyclops type of response in this particular pathway. We've also used this looking at particular, as I mentioned, genotype-driven effects. And if we look at the DIPGs, we now have some idea of drugs that particularly hitting in the H3K27 mutant versus wild type. And we do this both using isogenic knockouts of this allele, as well as other cell lines in the category that have wild type status. And in general, we're finding that ALK inhibitors particularly work better in the context of the wild type allele. And I think that's been previously reported. We have a number of other hits across this category. Just to go into our progress with the CRISPR genetic screens, I'm sure everyone's aware of how CRISPR works, gene knockout technology, and we're using it in the pooled screening platform. Our first generation screens, which are nearing completion, we should have about 100 cell lines completed in the next few months. We're currently at around 80. We've performed oncology drug target screens, focusing on about 350 genes that are either known targets or in the cancer related pathways. In the next, we've actually already started this in the past month or so, but we're really ramping up now on our next generation screens, which will include whole genome as well as combinatorial gene knockouts. So what have we learned from these kinds of analyses? Well, I think, you know, the ATRT cohort is a really good illustration. There have been a couple of papers focusing on CRISPR screens in ATRTs, but the number of cell lines was really not high enough to get down into the specific subtypes. And I think, you know, with this kind of cohort that we can now really have enough cell lines in the different subgroups of ATRT. And you can see here's group 1, which is mainly driven by the hedgehog pathway, and group 2, which is the MYC-enriched pathway. And we have a cohort of about 10 ATRTs in each cell line, which allows us now to look at subgroup-specific dependencies, and you can see those here. It was nice to see that MYC was particularly dependent in the group 2, as one would expect, and we're starting to see in the group 1 particular epigenetic dependencies that have not been described as to date. So the last part of this is really how can we start to do these analyses more systematically and share this with our colleagues. And as part of that, we are developing a data portal that allows us to share this data. In the last meeting, I talked about the drug data portal. We've now developed also a CRISPR data portal that allows us to look at the genetic dependencies and developed additional layering onto that that can enables us to look at the correlations between the different omics factors. And I'll just briefly show you a snapshot of that. So this is what the overall opening page of the portal looks like. You can see the number of cell lines that have been CRISPR screened, and you can see this heat map shows the different genetic dependencies across the lines. In orange are core essential genes, and you can see this just shows that the screens are working. You would expect the core essential genes to hit across all lines, which they do. And then if we just go through this, you can see the cell lines here. And we have two different views, a sample view and a lineage view. If you go through, I just can get to the sample view shown here. So you can look at a particular line. And I think one of the nice features is that we've also integrated the Broad Institute's dependency map. And you can see if you correlate the genetic knockout across all the dependency map lines, you can see that the ones in green score across, these are really the core essential genes that score across every cell line. So these are just core common essential dependencies, but focusing on this quadrant here really allows you to identify genes that are specific dependencies in this particular cell line or sample. And this is a CBTN sample, I believe, an ATRT line. The next part of it is the lineage view. And with the lineage view, you can start to look at particular dependencies in lineages. And you can see here in the DIPG DMG, you can enter this category and it shows you the top dependencies across all the DIPGs. You can see PIK3CA actually ends up being a very specific dependencies across the DIPG cohort. We can then look at the PIK3CA mutation status and correlate the dependency to whether that correlates to PIK3CA mutations. And the last part of it is really looking at whether PIK3CA drugs, targeted drugs, are showing a similar response. And you can see that here when we enter PIK3CA as a targeted dependency, these are all the PI3 kinase inhibitors. The ones shown in red are actually showing activity across the DIPG cohort. And you can see that indeed about 5 different PI3 kinase inhibitors show activity. Just to contrast that, if we were to enter a different drug, we would be able to see that this is a specific effect to the PIK3CA dependency. So just to summarize all of this, our program is now banked about 200 different unique pediatric brain cancer models. And importantly, these are models that actually grow well in the lab, and we are well underway to characterizing all of them at a molecular level, as you've seen. We're now partnering both with colleagues at CBTN and at the Broad to really, I think, begin to conduct bioinformatic analyses to identify therapeutic vulnerabilities. A few of these have already been translated into hypothesis-driven projects, and I think that's really the key theme of our program is really to generate new hypotheses and to work with everyone here to work at the lab bench to validate some of the targets and also to translate these into clinical trials, which we're beginning to do with the PNOC group. So with that in mind, I'd like to thank both the team members, but also in particular thank everyone at CBTN who has really been instrumental to build this atlas. This really requires a multi-institutional effort, as no one site can have the number of samples required to build this kind of atlas. And we look forward to working with you all in the future to expand on this. I'll take any questions at this point.